Hello, everybody, and welcome back to BioSC 140 Human Physiology. This is your video for Laboratory 3. In Laboratory 3, we're going to do measurement of plasma concentration. This is FOX 2.1, Laboratory Supplement, Laboratory 3. This is the first lab where if we were in person, we would be actually using our various students' blood for the procedure for the experiment. Now, we're online, so we won't be actually using each other's blood, but I still want you to be aware of the protocols involved in getting the blood sample safely. So, universal precautions. Universal precautions means you assume everything is hazardous. You assume that all blood is hazardous and you treat it as such. You wear gloves when working with blood of another individual. If you are working with your own blood, it's kind of hard sometimes to get a blood sample through a glove. So it makes sense, if you're working with your very own blood, you don't need to wear gloves, but when working with somebody else, you got to wear gloves. Dispose of lancets. So lancets it's a tool that kind of pokes the side that in lab, we use to poke the side of our fingers to get the blood sample. It looks like a little rectangle. It's like a rectangular box. And you put the box next to your finger and it has a spring with a little knife, like a lance on that spring. And so you put it next to your finger and bam, that knife comes out very quickly, punctures the skin, goes back inside the box. And there's a small, you bleed a little bit, allowing us to get a sample. Dispose of soft contaminated materials in the soft biohazard bag. Dispose of hard biohazardous material in the hard biohazard bag. Wash reusable equipment. So getting the blood sample is really the hardest part of this experiment. The first part, you start by warming up your hands. Warming up your hands will increase the blood flow to your hands. It makes this process a lot easier. Sometimes people will use the lancet on their finger and very little blood will come out. Getting your hands warm will help it. You then want to clean the area with an alcohol wipe, and then you use the lancet to poke a hole in the side of your finger and allow blood to come out. Usually we do it on the side near the tip of a finger because there's no calluses and it just works the best. You then kind of let the blood kind of pool on top of your finger. It's usually like a drop of blood like you see in this photo right here. And then you take one of these capillary tubes and you lightly touch that drop of blood. And the blood will actually move up into the tube. We then, once we have the tube full of blood, we want to put a cap on it. And to put a cap on these tiny capillary tubes, we use clay. It's actually this thing called critosseal. It's like a clay tablet. And you stick the tube into the clay, and a little bit of the clay goes into the tube and kind of seals one end. We want to collect 4 mostly full capillary tubes for this experiment. After we have the capillary tubes, we want to centrifuge the samples. Centrifuge them for 3 to 5 minutes. We want to make sure that the clay ends are on the outside of the centrifuge. The centrifuge is going to create centrifugal force, which is going to push the blood outward. It's going to push blood towards the side of the centrifuge. And so you need to make sure that the clay ends, the stopped ends, the blocked ends of the tube are on the outside so your sample doesn't go flying all over the place and get everything all dirty. You want to remember where your samples are and then let it spin for 3 to 5 minutes. What's going to happen with the centrifuge is your blood has a few different components. Your blood has a liquidy component called plasma, and then it has the cellular component, the red blood cells and the white blood cells. Centrifuging these samples is going to make all of the red blood cells move to the outside, move to the clay side of the test tube, of the center capillary tube. And you're going to actually see two distinct colors. You're going to see the red band where all of the red blood cells are packed, and you're going to see kind of this yellowish, creamish color on top. That's the liquid part with all of the red blood cells removed. And it's that plasma part that we want to use for this experiment. So we're going to take a razor blade and we're going to cut the capillary tube right above that red layer that signifies all the red blood cells. And then we're going to have to kind of coax the plasma out of the tube and into a sample dish. A lot of students, this is the first time they've used micropipettes. Some students, this is the first time they've used micropipettes. Micropipettes allow you to move very small and very specific, very accurate amounts of liquid from place to place. They look like this, and they have a button on top, a plunger on top. And if you push the plunger down, it goes down very easy to begin with. And then you'll hit a spot where there's some resistance. You can actually continue to push through that spot of resistance to within a spot where there's a hard, full stop and it's impossible to push it anymore. What you want to do is push the plunger down to where you feel that first set of resistance. That will allow you to pull up your desired amount of liquid, the amount of liquid that you've calibrated the pipette to pick up. Once you've pushed it down to that part, so first stop, this spot right here, you then put your pipette into the liquid that you want to pick up, and you release the plunger, you release the button. This is going to suck that specific amount of liquid up into your pipette tip. You then take your pipette over to the test tube that you want to put the liquid into, and you push down through the first stop all the way down to the second hard stop, and that will expel your desired amount of liquid into the tube, and then you can get a clean tip. Perfect. It really takes practice. So once you're able to use these live, you'll see what I mean. It definitely takes practice. All right, so we've gotten our plasma. We have our plasma now. And we're going to use something called a spectrophotometer. A spectrophotometer. Break the word down. So photo is like light. Spectro is like spectrum. Meter is like measure. So it's going to measure a specific part of the light spectrum. It's going to measure a specific part of the light spectrum. All right, so we have our plasma. And what we're going to do is we're going to prepare a sample in one of these things called a cuvette. The cuvette is going to have a couple sides that are frosted that are a little limiting for the light to be able to pass through. And there's going to be two sides opposite each other that are very clear, that are going to allow light to pass through. And that's going to allow us to get the reading that we want to get. In fact, in person, when you use these for real, it's really important that you don't actually touch the clear sides, because any dirt or oil that's on your fingers might affect your overall reading. So, when you use these, if you ever use these live, make sure you only touch the frosted sides and not the clear sides that the light goes through. All right, so prepare the sample. Each cuvette gets 2 milliliters plus 20 microliters. So 2 milliliters of your reagent and 20 microliters from your sample, from your plasma. So micropipettes, they're very accurate for very small amounts. 20 microliters is a very, very tiny amount of liquid. And that's where we're using the micropipette in this experiment. So prepare cuvettes in batches, put the reagent in, but do not add any samples until the plasma is ready. All right. So once we have mixed the plasma and the reagent, a future slide is going to tell you about the reagents and all the different cuvettes we're setting up. But all the cuvettes have the same basic setup. We're going to mix and let it set for at least 5 to 10 minutes to allow, chemistry to take place. After waiting 5 to 10 minutes, we're going to put these cuvettes into the spectrophotometer. So we'll get into what exactly we're doing with the spectrophotometer in a bit. So let's look at what's going to be in our different cuvettes. So typically in lab, we have 6 groups for this lab. And we have 3 large tables in the lab room. One table will do this experiment trying to calculate concentrations of glucose. So 2 groups will be on glucose, 2 groups will be on cholesterol, and 2 groups will be doing protein levels. So again, those are the 3 things we're testing. We're testing for glucose concentrations in our plasma sample. We're testing for cholesterol concentrations in our plasma sample. We're testing for protein. These are the setups. These 5 on this slide are the setups for glucose and cholesterol. It's a little different for protein. So glucose and cholesterol. We're going to have 5 cuvettes for glucose and cholesterol. Tube 1 is the blank. It's called a blank because it doesn't have any of our sample. It doesn't have any plasma. Instead of plasma, it's going to have water. It's going to have our color reagent, and it's going to have water, no plasma. It's our blank, it's our 0, it's our reference point. Test tube 2 is going to have 2 milliliters of our color reagent. Oh, one thing, the color reagent is going to be different for glucose, cholesterol, and protein. So if we're doing glucose, this would be 2 milliliters color reagent for glucose. If we're doing cholesterol, this would be 2 milliliters color reagent for cholesterol, and so on. For the standard sample on cuvette 2, we're going to do 2 milliliters color reagent and 20 microliters of a standard. A standard has a known amount of your desired chemical inside of it. So our standard for glucose, we know, has 100 milligrams per deciliter glucose. Our cholesterol standard, we know, has 200 milligrams per deciliter cholesterol. We're then going to do the unknown plasma, which is what we get from students. So 20 microliters plasma from student, 2 milliliters color reagent. We're then going to do it for a lab-provided normal serum. So human plasma from a sample that we know is normal. And then an abnormal serum. So the lab will give us a sample that is known to be abnormal for plasma. Just a little heads up, what's the difference between plasma and serum? Plasma is the liquid part of blood that still has clotting factors in it, and serum is the liquid part of blood that has the clotting factors removed. We can kind of use them interchangeably for this experiment, but we'll get into the differences when we talk about the cardiovascular system. We'll cover all that stuff. All right, so 5 test tubes prepared for glucose, 5 test tubes for cholesterol. For protein, we're going to do it similar, but a little bit different. We're going to still do the blank, the water tube for protein, our reference point, but we're going to do 5 different standards. We're going to do 5 different standards. We're going to do a 2 grams per deciliter standard, 4, 6, 8, and 10. 5 different standards for protein. All right, so we've got our test tube set up, and what the spectrophoto, so we've got our test tubes up, and when we put the samples in the spectrophotometer, it's going to give us something called an absorbance value. An absorbance value. Absorbance value is, the name kind of explains it. It's going to tell you how much light was absorbed by the sample. I'm going to get into the details and the discussion about exactly what's going on, but the spectrophotometer is going to give us an absorbance value. Our standard, or our blank, we know the concentration per deciliter is going to be 0. Our standard for glucose is going to be 100. Our standard for cholesterol is going to be 200. And our standard for protein, we have 5 different ones. We have 2, 4, 6, 8, and 10. Unknown is going to be the sample from students. Normal serum is going to be a sample from the lab, that's normal value. And abnormal is going to be provided by me, a sample provided by me that's known to be abnormal. The absorbance value is your data from the spectrophotometer to calculate. So concentration of water we know, calculation of concentration of standard we know. We do not know the concentration for our unknown, our normal serum or abnormal serum. And that's what we want to find out. We want to find out the concentration for the desired type of molecule in our unknowns and the serums we were provided. And we're going to use something called Beer's law. We're going to use something called Beer's law to calculate it. So right here is Beer's law. So once we collect our data in class, students go back to their chairs and they use this ratio right here, Beer's law, to calculate the concentration of the student's plasma, the concentration in the normal serum and the abnormal serum that's provided to us. Beer's law is a ratio. It's set up like this. Concentration 1 over absorbance 1 is equal to concentration 2 over absorbance 2. You can rewrite this equation into something that's a little easier to look at. Concentration of unknown, so that's what we want to know, concentration of unknown, is equal to concentration of a standard, so that's our known concentration standards, times the absorbance of our unknown divided by the absorbance of the standard. So after we use the spectrophotometer, we should know 3 of these 4 variables. We should have the concentration of the standard because it was given to us right here. We should know the absorbance value of the unknown because the spectrophotometer told us. We should know the absorbance value of the standard because the spectrophotometer told us. So we have 3 of the 4 values in this equation, which means we can do some math and figure out our unknown concentration. We're going to be doing this for all 3 sample types, glucose, cholesterol, and protein. Now, you're probably wondering, why do we do 5 different standards for protein? We did 5 different standards for protein because that allows us to use kind of an old school method of calculating concentration of our unknown. With 5 different standards, we are able to create a graph. We're able to make a y-axis with absorbance values, an x-axis with concentration values, and then plot our standards on that graph. You need at least 2 points to draw a line. For cholesterol and glucose, we only have 1 standard, so we can't draw a line with cholesterol and glucose. But we have 5 standards for protein, so we are able to draw a line. This line should start at 0, 0, because we should have a 0 absorbance value for our blank, kind of by definition. And then once we plot our known standards on this graph, we can estimate the concentration of an unknown sample. So in this example, we put our unknown plasma into the spectrophotometer, and it said that there was an absorbance value of 0.7. So our unknown got an absorbance value of 0.7. So once we have this all graphed out for the known proteins, we go with our unknown to 0.7 right here, absorbance value. We follow it straight over until we hit the trend line, and then we go straight down. And that should give us a good estimation of our unknown without doing any math, just plotting it on a graph. So 0.7 absorbance is equivalent to 3.5 grams per liter in this example. I'm going to stop this video here, and I will be back next time with the discussion video.